Hello students, in this video class let us start with the new concept, that is transcription, right? Transcription. In previous videos we have discussed regarding the mechanism of replication, right? So replication is one of the process which is conducted by the genetic material, as we know that DNA replication, right? So DNA can prepare DNA, means the duplication, the both strands are duplicated there. So by that the new two daughter DNA molecules are formed, and as we know that that is completely formed, completely along the length of DNA, how much it is there, that much means, so total DNA is become doubled there, right? So but in transcription it is also a copying mechanism, which is also copying, right? So here what happens, in simple words we can say that, so in a definition we can say transcription is a process, right? It is a process of formation of RNA from a single strand of DNA, from single strand of DNA, right? So it is known as the transcription, process of formation of RNA, right? So RNA as usual single stranded, which is formed by a single strand of DNA, not both strand it is using, right? So from that single strand, for example, see, this is what the DNA is there here, you can consider it as a DNA. So this the length may be more, right? So a portion of this one, a segment of this is what we call a segment of the DNA. See, so this segment, so undergo which is get opened, and so through which the one strand can synthesize the complementary strand, which is referred as a RNA here. Only one strand is copying. One strand is copying here. That means it is copying a mRNA, also called, it's a phenomenon, or so in other words, so in simple we can say DNA produces RNA. So this phenomenon is called transcription, right? Transcription, understood? So DNA can synthesize the RNA, but not both strands. In transcription, so only single strand is formed with the help of single strand. That is also said to be complementary strand of a coding strand. Here, for example, if it is considered as one is coding strand, another one is that is referred as a non-coding, or referred as complementary strand, right? So that is complementary strand. From this complementary strand, another strand is newly synthesized, right? So this strand is said to be the RNA, right? So this RNA is formed by single strand of DNA for a segment of DNA, for the segment of DNA, not whole the DNA, which how much it is required, or a particular gene. When you consider segment of DNA is called gene, so that gene is transcribed, right? That phenomenon is also referred as transcribed. The process is known as transcription. So this much of segment is transcribed into RNA, right? So this is what the DNA segment is transcribed to form a RNA, which is single stranded, right? So this is in the simple words, and also we studied this in previous video, that is central dogma. Once again I will recall here, central dogma, which is proposed by Francis Crick. Yes, Francis Crick, Watson and Crick, no? So one of the what you call the double stranded DNA model was explained. So Francis Crick and James Watson, right? So Francis Crick were explained or made a principle regarding this one. So that is central dogma we called, that is a principle or a set of principle or a rule we can say. So here the DNA can synthesize. So the DNA can synthesize DNA. So this process is called replication, right? Replication. And DNA can synthesize, yes, DNA can produce the what you call RNA, especially we call it as mRNA. So that is the process is known as, yes, transcription, right? So this transcription is the, and next one the translation. RNA can produce the, with the help of other this thing amino acids, usually tRNA and including this mRNA, right, ribosomal RNA, all are involved in this process that is called formation of polypeptide strand, that is called protein. So protein is formed ultimately. This phenomenon is called translation, right? So this is what we called the central dogma. This is called the central dogma. So we should remember, because the protein synthesis will follow this method. So protein synthesis totally depends on this phenomenon. This is what we call that as a rule. So in molecular basis of inheritance, this is a very important point. So that is central dogma, which is proposed by Francis Crick, you remember, and this is what we call central dogma. Means here the formation of RNA from DNA and the total protein is finally synthesized by a process called translation. Process of synthesis of protein with the help of this RNA, that is translation. And so this RNA can copy the DNA, yes, which is copied from the DNA segment, part of what you call DNA, part of DNA which is said to be a gene, right? So from that it is copied, right? So this is synthesized from the DNA, right? So where the site of synthesis of protein is takes place, ribosomes. Ultimately it will carry up to the ribosomes, mRNA, messenger RNA we can say, it is said to be mRNA. And one thing, so here also we can understand by this central dogma. So we can also write here, so if it is sometimes, so RNA can copy the DNA, yes, RNA can produce DNA, RNA can produce DNA. So that is called reverse transcription. What we call reverse transcription, right? So this is the reverse transcription we can say. So this reverse transcription is said to be, that is a reverse to the transcription, that is called reverse transcription. By that RNA can synthesize the DNA. So in some viral what you call HIV virus, so where it is takes place, right? So how we will see that in the particular video. Okay, so you should remember this central dogma. Okay, if you remember this one, so we can understand the remaining part, right? So that is the transcription. Let us we see the transcription in detail, right? Yes, see here students, see the process of formation of RNA from a single strand of DNA, from the single strand of DNA. Of course DNA is double stranded, so but only from this single strand, and one strand is copying, yes, one strand is only copying, another strand is coding strand we can say, so which is not copying, right? So another definition we can give, the process of copying, yes, process of copying genetic information. What it is copying actually? Genetic information. As we know in the nucleus, see if you consider this is the nucleus, so there is a DNA, right, which is double stranded DNA, right? So this double stranded DNA, yes, which stores the genetic information. As we studied, DNA is the better genetic material which will helps to store the genetic information, which is the better for rather than, yes, uracil. We have studied the differences, right? So which is a suitable genetic material because which stores the genetic information which is needful, right? So that is what the genetic information which is copied, right? DNA will not come out from the nucleus and it is not directly involved in the protein synthesis, right? So it can direct the protein synthesis. We can say that it is directing the protein synthesis where it is present inside by sitting there in the nucleus only, right? It will not come out. So instead of that, the messenger RNA is formed. Actually this RNA is referred as mRNA. mRNA is formed, right? So mRNA can get the copy of this genetic information, right? So it will copy from one strand. So even here two strands are there. So one strand which is said to be complementary strand, or we can say that the coding. So after the study of what you call transcription unit in the next video, we will understand that. So here for example I will take here two strands are there. One is said to be coding strand, another one is called complementary strand. So from this strand, so usually the RNA will be formed here, right? So RNA is formed here. Okay, so both these are the DNA strands. So this is what the RNA strand is formed. Later on we will understand that in the next video, right? So when you open strand, this is what the open strand what I have taken. For example, if it is third end and this is the fifth end and this is fifth end here, this is third end. So opposite to that, so this is become complementary to this strand, right? Coding strand is free here. So for this complementary strand there is a formation of a new strand, that is RNA, right? RNA strand is formed. So here that is copying, the process of copying genetic information, from one strand of DNA, from this strand of DNA, right? From complementary coding and complementary strand, right? One is said to be coding strand, another one is said to be complementary strand. So from this complementary strand, so it is copied, the RNA, newly synthesized RNA is copied. Only single strand is involved in the copying this during this process. That is what the transcription. In transcription only single strand is involved, but in case of replication both strands are involved. So there was formation of a replication fork. So one is become leading strand, another one is become as a lagging strand, as you studied in previous video, right? So here only one strand is involved. Why not two strands are not involved? So there is a question arises and simple answer will be there. So that is what if both strands are formed, what would happen, right? There is no protein synthesis, there is no use of transcription, right? So for the transcription in detail let us see that. So but you should remember this is important. So what is transcription or define transcription if they asked? So you can write this point, okay? Process of copying genetic information from one strand of DNA into RNA, right? One strand is involved in the copying process, that is of DNA, and by that the RNA is formed, right? So this is the, and during the process the principle of complementarity what we call. So during this process, this what we call transcription, complementarity, which is what we call Chargoff's rule. Complementary means what here? The Chargoff's rule will be, yes. So as you know, Chargoff's rule means one purine is paired with another pyrimidine, that is A always pairs with T, right? So and in case of DNA, in case of replication, A pairs with T and guanine pairs with uracil by triple bond. So this is what the complementary, it is said to be complementary. But in case of this is of course it is applicable to the, yes, in replication process, but in case of transcription their RNA is synthesized. What is synthesized here? RNA, right? So during RNA only, instead of this adenine always pairs with here thymine in case of replication, but here what happens? So that would be yes, uracil, right? So A pairs with uracil while in the preparation of RNA, right? So in the same way guanine pairs with cytosine, no doubt. So this is what the complementarity in the transcription. In transcription, this is in replication. During replication, A pairs with T because DNA synthesizes the DNA. DNA can prepare the DNA there. So, but in case of transcription, DNA is preparing RNA. So, here the instead of thymine, there is a uracil is pairs with adenine, right. So, during the process, so that's what the point here. During this process means in the transcription, so the principle of complementarity governs the process, that process of transcription. So, except this, right. So, same replication, how the replication takes place there. So, the same way here also in the transcription also it is same, but except this one, adenine pairs with uracil, not with the T. So, that is what I have written here, that is A pairs with U means uracil, not with the thymine. So, this is about the transcription. And why here only single strand is involved, only one strand is involved, why not two strands? Let us see that.